The Center for Cancer Research is an NCI-designated center that was first funded in 1978. We're a basic science center, one of only 7 in the country. And so our focus is really on understanding how a cancer cell works, what makes it a cancer cell, and then developing drugs to interfere with that process. So as we identify specific pathways, we develop small molecules to impact those pathways and block the signaling process. So that's more of our chemistry side in developing these. We have a very strong engineering component. We develop a lot of instrumentation, unique imaging processes that really build on the discoveries of the cell metabolism.
A lot of times it's also Purdue investigators that say, look, we're working with a new type of technology. For example, we are detecting the earliest signs of a particular disease. Maybe it's an autoimmune disease, or maybe it is cancer, where we, through some kind of mass spectrometry-based analytical method, we can look at the telltale signs that cancer is going to start occurring or that cancer progression is happening. And I know that we have those tools here at Purdue, and the investigators that are developing those tools are looking for clinicians that could put these types of analysis platforms to a test and really find that clinical relevance.
Our lab is called the Purdue Proteomics core facility. So it's a core facility. It means we provide the technology platform for the investigators in West Lafayette campus, as well as the investigators from outside in the state of Indiana, even beyond. So our capabilities are to identify the protein, and we can quantify them. So then we can measure how these proteins are regulated in the cell. We can also characterize those, how this post-translationally modified protein, how the proteins are phosphorylated under different conditions. And many proteins, they function by interacting with other proteins and other cellular components, like not only protein, but they also interact with the small molecules and the lipids. So we provide the platforms or the technology so that the investigators can come here, or they can sample, and we can map these modifications, as well as we can map their complexes, how the one protein interacts with the other protein to execute the functions. The other aspect of our work is to support in the data analysis pipeline too, because these are the very large scale data sets. We support them by analyzing the data, mapping those data to different pathways.
So I'm in the flow cytometry core facility, and what we can do is analyze huge numbers of cells in a very short amount of time. And we can take something like a tumor, human tumor, and instead of being made of just one kind of cells, human tumors may have many different kinds of cells in them, and some of the cells are dead and some of the cells are alive. And we can help researchers pull apart these different cell types and understand what's the contribution of many different cell types. And we can actually physically separate them in the cell sorter so that we can hand back purified cell populations. We have a couple really outstanding pieces of equipment with our ARIA cell sorter and our Fortesso, so we can look at up to 18 different parameters at the same time.
My lab for the quantitative analysis uses mass spectrometry. We get all different types of tissues. We get not just blood, but liver, kidney, hair, you name it. We can quantitate the pharmaceuticals in those different matrices. And that can be challenging because now we're dealing with thousands of compounds in the background, and we call that the matrix. And you're just looking for one, so it's kind of a needle in the haystack experiment. But our laboratory has many types of state-of-the-art mass spectrometers. So depending on what the analyte is that we want to look at, it dictates on what instrument we use. We will often get samples for what we call comparative analysis. So we'll look at, say, a plasma sample prior to the drug being administered, and then we'll look at the same patient who has given us plasma samples post-administration of the drug. And we're setting the instrument up this time not to be specific just on the drug, but really to look for as many molecules as possible. In a biological sample, we can see thousands with our instrument in just about a 30-minute analysis time.
When people ask what is what we do, we say, I usually say we're a bioanalytical hub. If you have a sample, we'll give you analysis. And specifically what we do in the lab I work is mass spectrometry. And in this technique, small molecules, and in my case, the lipids, we look at different classes of lipids, many classes in a very fast way. So in this method, Purdue has IP on this method, and as far as I know, we're the only facility that offers this type of very fast exploratory studies in lipids. We call this accelerated biomarker discovery of lipids.
So I'm in the biophysical analysis laboratory, so we focus on characterizing molecular interactions with biophysical instruments. So the interactions including protein self-interactions and heterogeneous protein-protein interactions, and other interactions like protein binding to small molecules, protein binding to DNA or RNA, DNA binding, nucleotides binding to small molecules. We have state-of-the-art instruments to characterize interactions. We also have expertise to do the data analysis. So combining the instruments and the data analysis, we can put together a full picture of the molecular interactions.
Our imaging facility is really broken down into two different categories. One is the preclinical imaging techniques or instruments, and the other one is microscopy. It spans the whole resolution spectrum, from as small as down into the nanometer range all the way up to the whole animal. Within the preclinical instruments, we have nuclear medicine instruments. We have an integrated SPEC PET CT instrument. We also have a micro CT for in vivo animal imaging. And the last instrument we have is the bioluminescence or fluorescence instrument for whole animal imaging systems as well. On the microscopy side, we focused on confocal and super resolution instrumentation. Not only do we have a wide variety of instruments, but we also are highly technically capable to teach the users and teach the researchers on how to use those instruments, how to collect their data, how to analyze their data, so they can then move very quickly on to their next project or next question in their project.
We are on a time where analytical technologies, mass spectrometry, can do a lot for translation, and we can see that. If we can do this molecular investigation inside the clinical trials, we think we could see a lot of, what makes people to be good responders, and can we see people that will have side effects? Can we relate clinical data with metabolomics data? So this is really what we are trying to get into, and I think we'll start seeing more and more of that. We can get relevant samples, patient samples, very good clinical data, control environments, and we can apply these biomarker discoveries, and try to find out, good responders, and any information that we can have that these people would benefit from.
We work with people all across campus and people across the state that need our particular technical expertise, and our work bridges from very, very theoretical beginning, cutting edge, let's just try and see if something like this can work, to actual clinical direct application.
The research means is always interdisciplinary. Yes, you can bring the people from different backgrounds, so we have the different expertise together. And when I reach out, everybody appreciates that, and we always work as a team. That's a win-win situation for everybody.
The efficacy is very important, and the safety is very important. And how to improve the efficacy, and then how to design a new drug, and then you need to know whether drugs interaction with the target. So in my facility, we have people to understand the interaction between a drug to the target, So if you know the drug to the target interaction, you might develop a new drug. You might just improve the efficacy of the old drug. At the BAL, we have instruments help people to understand the formulation, how this formulation affect the drug's absorption in human body.
My laboratory is in Discovery Park, which is a certain section of Purdue that really was designed to foster collaboration between different departments or different disciplines. For example, I'm working on one project right now where the samples come from clinicians down in Indianapolis, so we have the physician aspect down there. I get the sample, so I'm an analytical chemist, and I'm doing the quantitation of the drug as well as looking at some of the other molecules that are perturbed when a patient takes that drug, because this particular drug is known to have some side effects. That analysis part is very complicated, and we've collaborated with chemical engineers who like to do a lot of computer modeling. So here we have the physicians collecting the samples, I'm a chemist doing the analysis work, but then the data interpretation goes to chemical engineers to understand what's going on. So many different groups have come together for this one project.
Purdue is an absolutely great place to do science. This is a place where people work together. I've been in a lot of institutions where there's so much competition and just fear between individuals that people are selfish and don't want to interact with other people. They don't want to tell you, what they're really doing. They're, just how can I get a little bit extra piece for my own pie? And here people really want your research to work.
We are a basic science center. We don't have clinical outlets. And so being able to engage the Big Ten clinical consortium provides us with those outlets. We right now have 11 drugs that are synthesized here for cancer-specific therapeutics that are in clinical trials around the country. And we have many more in the pipeline. And so what we would like to be able to do is to take those drugs and move them into investigator-initiated phase 1 trials to begin to validate them in a more rapid way. And we think that this consortium is the perfect approach to doing that.
It's important for all of us to work together across different institutions, because doing cutting-edge science or cutting-edge medicine is tremendously expensive. And it only makes sense if you're going to have equipment like that you share it with other people and you make sure that equipment is used to its fullest. Because otherwise, if it's just sitting there, then all it is like, a piece of junk, really. It has to be busy to be useful. So the more people can bring us samples, the happier we are.
So we're working on the medicines of the future. So we know in 5 or 10 years what we're doing now is really going to have an impact, and it's going to be cutting edge.
We're all committed to making treatments better for our cancer patients. And while we study the basic biology of cancer and try to understand how these cancers really become malignant, our overall goal is to help the patient. So we've got to get that knowledge, that basic knowledge that we discover, to a point where it can help patients, and we're all committed to doing that.
I'm very hopeful that we'll be able to contribute, as every research dreams about, doing a positive effect on the society. That's the best trophy we can get. It's to know that our work really impacted positively the life of even one person.